if we were to be working up that little organization chart, the atoms up to a protein, up to a cell, up to tissues, organs, and so forth, we'd be moving on from the chemistry up to the cell. Now, the cell has a lot of things going on with it. Within it, you have little things called organelles. If you think about it as a human being almost, the little things inside of it would be kind of like the organs, the functional pieces of the cell. We're going to discuss in greater detail about these organelles in another part, but we're going to be discussing most importantly that of the plasma membrane and what it does. Let's hit some of the basic structures really quick. You got the plasma membrane, you got the cytoplasm, which is the space within the cell that contains all the liquid as well as the organelles floating around. And within that cytoplasm, you also have the nucleus, which houses your genetic information, what makes you you. The plasma membrane pretty much keeps you from a cell being a cell rather than just a homogenized liquid throughout the bottom of a sea floor. It's flexible, it's sturdy, and it's made of fat. Now, as a lot of y'all are aware, if you're a cook, if you're a chef, you can have fats that are liquid. And in fact, this plasma membrane is quite liquidy. These little proteins, these little things that are on the outside of it, actually are moving around quite a bit. It's a fluid, but they're staying right there because they really, if you remember back, the hydrophobicness doesn't really like hydrophilic stuff, meaning the stuff that doesn't like water doesn't want to really hang with the stuff that likes water, so that keeps them bound, these proteins bound where they are. But they sure do float around in this thing. Now, in it, you have a lot of different proteins that can do amazing things. It can let stuff in and out. It could identify who it is. It could be a lot of things. Let's go ahead and just label out what these things are. For the very first one, you can notice up in the upper right, you'll see that there's an ion channel. So most ions are not able to cross over through a plasma membrane willy-nilly by themselves. Only water is able to do that, surprisingly, and oxygen. And maybe not carbon dioxide. But they have to form a pore to let those ions in because they have a charge on them. Hydrophobic stuff usually doesn't have an electric charge on it. It doesn't like charges. It doesn't like positive, doesn't like negative. It's a neutral entity. So you have to make a pore for certain ions if you want them in that cell to be open and available. You can also have carriers, meaning that you only want a certain amount of really bulky items such as sugar, some amino acids, or things that are important can be transported in, and it's sort of like a I don't know if y'all have ever seen a submarine door and how it operates to depressurize or any type of decontamination zone. You enter into a room, the gate with two gates. You go in one gate, that one gate closes behind you, water excretes out or what have you, and then you're released out into the wild or released out into the submarine or what have you. That's what a carrier kind of does. A receptor, oh boy, you got a lot of those. Receptors are proteins that will react with little chemicals on the outside and cause the cell to change. So for instance, you got growth hormone, you got insulin that would interact with these receptors. You got pretty much any endocrine molecule that's water-based will interact with a receptor like this. You also got enzymes. Special cells could have an enzyme on the outside that changes or breaks down stuff that's floating out and around. You could also have linker proteins which help keep the integrity of the plasma. Maybe it's going to link up with another cell to make some tissue. You also have the cell identity markers. That's the "hello, my name is Bob" marker type of thing. It a lot to do with these like glycoproteins. These type of structures are a lot to do with how you determine who you are, as well as to see if there's any foreign material that your immune system should attack. Now, permeability, we kind of discussed it a little bit already. All that means is remember that it is nonpolar. It does not like charged molecules, so it's going to do its very best to keep that stuff out. Only way you're getting that stuff in is through some type of channel. Macromolecules will have to have some very large molecules, not just simple sugars and like that, but like proteins and things like that will need to be brought in via vesicles, a vesicular transport, which we'll talk about here in a little while. Another thing that really drives is whether or not how a chemical goes between the plasma membrane and using these channels is how concentrated it is. It could also be the electrical gradient. So for instance, potassium is a positive ion, but if there's a lot of positive ions on the outside and potassium's hanging out on the inside, it's really not going to want to go out as much. But it will, because its concentration gradient outside the cell is usually pretty low. So together, they're called the electrochemical gradient. The electrical gradient, chemical gradient, they're called the electrochemical gradient. Now, transport across the membrane cell and how it works. There's several ways that this could happen. It could be a passive process. Passive meaning like, sure, just let it happen. Nothing stopping it from occurring. Maybe there's even a channel open up. This is you need to know this stuff. That's the shooting star going, the more you know, da, 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 da. You also have active processes. That means that at the electrochemical gradient, maybe you're trying to fight against that. And that requires a lot of energy. So you're actually going to have to use some type of energy to make that work. Now, processes that go as a passive thing is simple diffusion, sort of like water passing through a cell membrane. There's nothing really stopping it. There's a facilitated diffusion, which is like the ion channels, which we talked about. It's just a pore in the wall of the cell membrane that just sort of lets things in and out. And then osmosis, again, that's this more specific term for the process of simple diffusion for water. Osmosis is the diffusion of water through the cell membrane. Active transports, you got the primary and secondary transport as well as that vesicular transport, which maybe you remember from back there a little ways. It's that when you have something massive you have to bring in or push out. Now, before we go on about this concentration gradient, I'm going to give y'all a tip on how certain meats are cooked. Well, how meats are cooked and how you can make it nice and juicy. I apologize to those that are vegetarians. Please just use this as an analogy. I'm not asking you to eat meat. Anyways, this is how when you put on a salt rub onto a slab of meat, you have to let it rest for hours, or else it's going to turn out really bad. It's going to come out dry. It's going to taste gross. But after hours and hours of letting it marinate with that rub, the meat becomes really juicy and falls off the bone. Any idea as to how? Well, we're going to go ahead and go over a few things on how this works, diffusion, how osmosis works, what is osmolarity, and that should help determine and help you know how this happens. Now, first off, let's talk passive transport. Remember that this is going to be a simple diffusion. There's nothing barring it. There's nothing getting in the way. It just happens. If you have a high concentration, that high concentration is going to want to go towards the low concentration. Kind of like you see it out in this corner here. You can see that there's a very high concentration of some type of blue liquid that over time, that blue liquid is going to become less and less concentrated and dilute across it. It diffuses. There are 5 factors that determine how quickly this diffusion occurs. Now, I went ahead and I likened this to trying to get 2 different types of individuals going through a little McDonald's playset or some type of happy go pukey playset. One, it could be a little small child that's 5 years old, nice and scrawny, or you could have an 18-year-old that is morbidly obese, 500 pounds, trying to get through this. So the travel distance, if you're a small kid, this is pretty easy. It's going to go really quick, but if it's long, it's not going to work that great. In temperature, if it's really, really cold, no one's going to want to go. It's going to go a lot slower, but if your temperature's high, you're going to move quite nicely. concentration, if you got a line of kids trying to go through, they're going to be pushing on each other to get each other through. While the viscosity of the media, if you have this filled with liquid, those kids are going to go a lot slower. But if it's nice and thin like air, then it's going to be like, bam, all done. Well, the size of the diffuser, that's the comparison I was trying to make with the large person versus the small person. That small kid's going to have a really easy time getting through those tubes compared to that 400-pound 18-year-old. So those are the factors that affect diffusion: travel distance, temperature, concentration, viscosity of media, how thick is it? Is it like maple syrup or is it water? Or size of diffuser, is it a small child or a giant 18-year-old 400-pound man or a woman? Now, with that, let us talk osmosis. Osmosis is water's tendency to follow after salt. So here's an example of what they're trying to do, how to explain it a little. On the right side, or this right arm of this flask, we've put water in it. And then we add salt to this side. What you'll see when we do that, and there's a membrane right in the middle that prohibits these solutes to cross over to the other side. So what happens is water really, really, really likes charged stuff. It really likes salt. Just absolutely adores it. So water will actually go towards the salt, cross this membrane, and the liquid will actually go and try and dilute and become equal parts concentrated. Now, let's take that into account into biology. So we got ourselves a normal cell. We got an isotonic solution. Isotonic means that the salt concentration on the outside of the cell is the same concentration as the inside of the cell. Looks nice and normal. There's a red blood cell. It has a biconcave disc that looks like a little inner tube. But what happens if we were to take the same red blood cell and put it into a liquid that has little to no salt in it? Well, we throw it in, and all of a sudden, the water that has absolutely no charged friends to play with finds it inside the red blood cell, and it migrates heavily into it. So that water just charges right into that red blood cell, and that cell just doesn't have a chance to push out that salt, and it becomes very bloated, and it could lyse. Lyse means to pop or break. So hemolysis is the breaking down of blood. Now let's say we take another cell that's a nice normal cell, biconcave, looks like an inner tube red blood cell, and we drop it into a hypertonic. Hypertonic solution is when we have way, way too much salt. Way too much salt in that water compared to a cell. You drop it in, what happens to the water inside that cell? Well, that water goes, hey, I have a lot more friends on the outside than I do in the inside. So it runs away out into the solution. It gets sucked up and absorbed by that salty solution, and your cells begin to crenate. That means that they start forming and looking weird because all the water has gone out of it. Those of you that are nurses or going to be nurses, what you are trying to do when if somebody comes in dehydrated into the ER, into your clinic or what have you, what you're going to do is you're going to give a saline solution. It's an isotonic solution to your blood. That way you don't pop it. If you put in pure water, which would be your hypotonic solution, you're going to make some uh-ohs and boo-boos, and that person is not going to end very well. If you put it in over salty, well, you pretty much defeated the purpose of not making the person dehydrated. You're making it worse. So that saline solution is to mimic your blood and make it isotonic solution to your blood plasma. Now, with that, let's go back to our little thought process. What happens to the water in the meat at first? So you throw salt on it, and all of a sudden the liquid from the meat is going to outpace the diffusion of the salt, and it's going to go inside, and it's going to get leached out of that cell. So if you're to throw that meat, you do the rub, and then you put the meat onto the grill, you're going to leach out that liquid, and you're going to cause it to pretty much evaporate as soon as you hit the grill. But if you let it sit for hours on end, that salt will eventually seep in, slowly diffuse into that meat, and that salt will actually hold on to that water much more readily and keep that meat juicy. So there's your tip of the day. All right. So with that, let's continue on with the passive transport, aka let's go via facilitated diffusion. We've talked about osmosis, which just happens readily. We talked about passive diffusion, which just happens readily. We're going to talk about facilitated diffusion. It means that the molecule or the ion that you want to cross really, really wants to cross, but it can't. And the only way it can cross is if you give it something. You facilitate it. You make it possible. That's what facilitated means, is to allow something to become possible. So there's two ways that you can facilitate diffusion. You could allow for a channel and the thing just leaks in. You could also have a carrier, but the carrier will have to bind and do like an open close thing, like that submarine thing I talked about earlier. Now there is a way that you can actually, these pores can have a gate, so that way you don't lose all your potassium out into the wobbly yonder if you want to hold on to it, or whatever molecule you want to. You can the cell can open and close it based upon the demands that's on it. Now a carrier mediated is that submarine thing again. Something comes along like glucose. Glucose is a carrier type thing. It binds to the inside, stimulates the reformation or change in the shape of the cell. It's a conformational change is the appropriate term. And it allows, by doing so, it allows glucose to get in, along with the glucose concentration gradient. That is passive transport. I'm going to go ahead and pause there, and we're going to go ahead and make the active transport discussion a discussion in and of its own.